Our next presentation is by Professor Linda Columbus from the University of Virginia. And Linda has been one of the leaders in the area of membrane protein NMR studies by NMR spectroscopy, and a great influence on our work too. And the title of her talk is The Structure, Dynamics, and Function of Proteins Involved in Bacterial Pathogenesis, LspA and Opa, or Opa.
Thank you. Thank you to the organizers for inviting me. And I'm just really feel honored and thankful to be a part of this. And thanks for the introduction, Guy. My lab definitely focuses on membrane proteins, and we have training in NMR and EPR, and we really just focus on trying to understand how some membrane proteins work. So that's my subtitle, and I probably should have just given that to Guy to make it easier on him. But you'll learn how it's Opa, and it stands for opacity. So I'm going to try to get 2 vignettes in, so I'm going to get right to it. The first vignette is on the Opa proteins, and it's work we've been working on this project for quite a while now. To start, both these proteins are not channels or transporters. So I'm the outlier, I think, in this membrane protein session, and so I want to give you proper background so you can follow what we're talking about. So one extremely interesting function in several bacteria, different bacteria, and these different mechanisms, is that they're able to induce cellular uptake in human cells. And they can do this in a receptor-mediated way, a lot like viruses. Some use similar receptors. In this case, I'm showing you Neisseria gonorrhea, which, at least in the lab, is predominantly a Diplococci. So you can see two red, falsely colored red bacteria being engulfed by a HeLa cell that's expressing a specific receptor here, and that's colored in green. I just love to start with this picture just because it just gives you this really powerful image of what we could do if we understood this process. We could actually gain access to cells in a receptor-mediated way if we understood how this one protein, so this function comes down to one protein, and it's called Opa. And Opa proteins are actually a family of proteins that are exclusively found in Neisseria, as far as we know. We've looked at a lot of Opa-like proteins in the sequence databases, and so far as we know, this is the function solely in Neisseria. And so lots of proteins bind to the receptors that the Opa proteins bind to, but they don't necessarily induce engulfment by the human cell. And so just showing you a schematic here of Opa proteins expressed on a Neisseria surface, engaging 3 of the possible ways that Neisseria gains access inside a cell. And that's through CEACAMs, which is exactly what I'm going to focus on today, with alternative pathways through HSPGs or syndicam 4s, and an HSPG-mediated integrin signaling pathway that will also induce engulfment. And so we're going to today only talk about the CEACAM interactions. And specifically, we're going to talk about CEACAM1. So CEACAMs are carcinoembryogenic antigen-like cellular adhesion molecules, and they are involved in a lot of cell-cell contacts and interactions in a lot of cellular processes. And they come in different flavors. And so what you can see here is in red, they all have an N-terminal IG domain. And that is what we're going to discuss today because that's what interacts with Opa. It's also what mediates the oligomerization. There's homo and heterotypic dimerizations between these CEACAMs. CEACAM1 is predominantly a homodimeric interaction between CEACAM1 and CEACAM1 across cells and within the same cell. So a trans and a cis dimer interaction to then signal. So it has a transmembrane helix and an ITIM domain on the cellular side. So CEACAMs can have ITIMs or ITAMs, and they can be GPI anchored. So with that, we'll focus on CEACAM1. Opa binds to CEACAM1, 3, 5, and 6, and doesn't bind to 4, 7, and 8. It's interesting because the N-terminal N-terminal domain of each of these receptors is quite similar. Structure's been out for a while. There's several structures of each of the different CEACAMs, and it's thought that this dimer interface is the one that is the trans dimer interface, which has not been confirmed, but is the leading hypothesis at this point. And the CEACAMs, because their ectodomains are actually glycosylated, and in the endomain, it's glycosylated in 3 different sites. And so the crystal structures, until recently, when my postdoc Megan crystallized the truncated glycosylated protein, which has the exact same dimer. And that's why this is thought to be the biological dimer, because those glycosylation sites would be sort of starkly hindering the dimeric interface. Opa binds at that interface. Before I go there, I just wanted to show you the glycosylated dimer, which is almost identical to the non-glycosylated dimer. We've also have extensive data in solution using DEER and other techniques showing that the truncated glycosylated and the complex glycosylated is dimeric in solution. So we think this is the structure that Opa would be interacting with, and we think it competes for the dimer because this is the face that's been mapped of the endomain to interact with Opa. So these are sites that would be right at that dimer interface. What's in red is the residues that are essential for interacting with Opa proteins, and in orange is different Opa proteins can have different binding maps onto the surface of CEACAM. So what do I mean by different proteins? So since they're all in Neisseria, what is this family? And so the family there are 11 alleles in Neisseria gonorrhea. There are Opa proteins and meningitis as well. There's 7 alleles. Within those, there are variable sequences. So at any given time, all or none of those could be sequenced because there's this phase variant expression. And within a colony, you can have different sequences within it. If you were comparing between bacterial colonies, between patients, let's say. So if you isolated an Opa protein from a patient, it would probably have different sequences that have not been characterized. And so this has been done, and over 338 distinct Opa sequences have been identified. And so what I'm showing you here is the regions that have been identified to interact with CEACAM. So Opa proteins are 8-stranded beta barrels. They present extracellularly 4 loops, 3 of which have been identified to determine specificity to CEACAM binding. Then the first loop is a semi-variable region, and between in loop 2 and 3 are 2 regions called hypervariable regions, and this is where the sequences vary. The barrel itself within the sequences is 94% identical. And so there's recombination that's occurring in specific regions of the allele to cause this variation. So that's from a molecular biology and structural perspective, that's an interesting question. How would you have these different sequences that bound the same receptors? And how is that determined? So that's the question we're seeking to answer. In addition to that, we want to be able to hijack this function and capture it in vitro in liposomes. And so I'm going to show you our progress towards this. So the first, to determine the answer to the question, the answer to the first question is, how do these bind to the receptor? We have to start looking at the structures. But before I do that, I want to highlight these different sequences so that the data I'm going to show you shows that these are dynamic loops that are in an ensemble, but they don't have the typical intrinsically disordered sequences that I'm more familiar with, because they have these loops have hydrophobic residues in them. So you'll see there's isoleucines, alanines, valines. There's lots of functional diversity in these sequences. And if you notice in HV2, there's at least 3 to 4 prolines within a 15 region of the sequence. And those positions are never conserved, but they're present. we don't know what to make of all this, but these are the observations for which, if you look at these sequences, now they're just too variable to be able to look to find motifs to say, oh, okay, if you have these characteristics, you will bind CEACAM1 and only CEACAM1. And if you have these characteristics or this motif, you'll bind all four. And so we need structures. Okay, so to start on the structure, we determined it with NMR. And we had a beautiful beta barrel and absolutely no restraints in our loops. And we have about 30% of it assigned with NMR. And so we worked with Peter Casson and used MD refinement to just get a sense of what the spatial distribution was of those loops. And what we found was, yes, of course, we have excursions of these loops in all regions of space, but there is an increase in the density of the loops more above the barrel. And then we can start analyzing this in terms of where is a more dominant interactions versus other places. But before we do that, we wanted to look at the dynamics. And so with NMR and molecular dynamics, these loops are dynamic on the nanosecond timescale. And it's classic polymer prediction in that the loops are more, the center of the loops are more dynamic than where they're attached to the barrel. And there's just a gradient in the dynamics as you move towards the center. And just to confirm that, because we only had 30% of the loops assigned, we went ahead and put spin labels every 10 residues in every loop, and we looked at it in both detergent micelle and in liposomes, and the NMR was done in micelles, and we also looked at nanodiscs and showed that the proteins were in the same environments, or under the same conformation in those environments. And in here, we see the same in the liposomes versus the micelle. We see this the same as we saw with the MD, this increase in dynamics as you come to the center of the loops. And in the EPR, we're not capturing any tertiary interaction that's either long-lived or stable enough to be seen with the EPR. So we looked at the contacts in the refinement, the MD refinement, to see if we could see if there's any preference for those two HV regions to come together. And so we knew from experiments that had done chimeras, if you take an HV1 and an HV2 from a protein that binds just CEACAM1, and you switch one of those with another protein that only binds CEACAM1, you abolish binding for both proteins. And so we knew you had to have a specific combination of HV1 and HV2, and that's had started to lead us to a hypothesis, which we're leaning maybe against currently, that HV1 and HV2 came together to bind CEACAM. And keep in mind that surface on CEACAM is quite small. It's 330 angstrom squared at most. And so we're probably thinking about maybe 6 or 7 residues on Opa that might be interacting with the CEACAM receptor. It also means that most of the loops are going to still be dynamic even in the bound state, just giving you the complexity of the situation from both spectroscopy perspective and a crystallography perspective. So indeed, we did see some higher contact probability between HV1 and HV2, but if you looked at the lifetime of those interactions, they were less than 5 nanoseconds. Okay, so it's a structure, and it's a dynamic ensemble. And so Peter and I are very interested to understand this dynamic ensemble in the way that, is it a conformational selection? Is there a conformer that is being sampled that could be recognized by CEACAM? Is there an induced conformation? What does this ensemble look like, and which, at the end of the day, which residues are interacting with CEACAM or even are available to interact with CEACAM? And so we started to want more information to refine this ensemble. And with only 30% of the loops assigned, so most of the dynamics, in addition to the nanosecond dynamics, we have some slow motion and heterogeneity that lead to line broadening, and so we don't see most of the residues in these loops in the NMR experiments. So we went to EPR and DEAR, and we engineered 2 spin labels and measure the distances between each of these loops. And now if these are random conformations, we're just going to get Gaussian distributions around these distances. And indeed, if we just keep picking random ones, we just may not get much information. So what Jennifer did was develop an algorithm to help us predict informative pairs for this ensemble. And so indeed, we got with her predictions and not with our selections, specific distances that now don't just have these broad Gaussian distributions that you would expect for a random distribution of states. And so these are these distances like the 86, 158 or 28 and 159 are the ones that are going to give us informed information about the ensembles. The nice thing about this technique is that we can just keep doing this in iterations. And so for now, the iterations we have given us a couple of states that are more populated than others. And so we can cluster them and start to look at them and come up with hypotheses about binding, which we're doing. We're not quite ready to share. And because we want to compare it to the bound state. And so we want to start doing this with the bound state. And using the glycosylated form of the protein, we can start to look at, and so in green is with CEACAM, we can look at these Deer distributions with and without CEACAM of the loops and see if certain populations that we've seen in the APO state ensemble are selected for. And if they are, we can continue to refine around those. So that's where we are at the moment. Functionally, I am completely behind time. And so I'm just going to say functionally, we've reconstituted these Opa proteins into liposomes, and we can characterize their uptake into different cell types, HeLa cells here that are expressing CEACAM. And we can show that those are dependent on the CEACAM signaling pathway. And so we're working on looking at these liposomes both with cell culture, but also in vivo with human neutrophils, and looking at the phenotypes for each of these OPAs and monitoring their uptake. So with that, I don't have a lot of time, but I want to switch into a different protein and introduce you to a lipoprotein signal peptidase that we've been working on called LspA. And I mainly picked these two topics because I really wanted to focus on two things. One is how structure informs in function and dynamics, and where the future is in structure in terms of how we might need to think about other restraints and information that we would want in order to define conformational ensembles and states. And so for LspA, we have another story. So LspA is the peptidase involved in lipoprotein processing in both gram-positive and gram-negative bacteria. In gram-negative bacteria, it's essential. And so it is an antibiotic target. And at the moment, there's no commercial antibiotic, but there are several known natural product inhibitors or antibiotics. And so Martin Caffrey determined the structure in 2016, and it's shown here. And he determined it with globomycin bound, so that's one of the antibiotics. And to date, we don't have any APO state of the protein. So what this protein looks like without the glycosylated form. I'm sorry, without the antibiotic formed bound. And so here you can see is where the globomycin is bound, but you can also start to see where the substrate might be interacting, where the helix is that gets cleaved, and where the lipoprotein would sit on top. And then he termed the two phrases beta cradle and pH, and I'm going to use those because, as you can see, they form this cavity that interacts with substrate in globomycin. So we started work on this because we wanted to look at the conformational states of LspA to see if we could start thinking about how we might target this protein with different antibiotics, and how maybe if we included something that mimicked the actual signal peptide, which would be the transmembrane part, how we might model that. And so we wanted to understand the conformational change with respect to that. And so we started looking at APO, and then we started titrating globomycin, and we decided to take a spin labeling approach. We just haven't been able to get enough yields to do NMR experiments with. So what I'm showing you now is there weren't a lot of residues that were sensitive to globomycin binding, but one of them was shown here in a yellow sphere. And what we found immediately is that our EPR line shape changes with titrating the globomycin in never saturated. So we got mobility changes, so the site gets more immobile, But then there's also another conformation that arises as we increase the titration. We were very confused at what was going on. Until, well, we still might be confused, but another structure that Martin came out with was of a different organism. So the first one was from Pseudomonas, and the second one is from Staph aureus, with globomycin bound and myxovirecin. And the myxovirecin structure has that beta cradle and pH. The antibiotic's still bound, but the cradle is more, the cradle and the pH domains have come together. And that's shown here by this helix it becomes more ordered and down onto the antibiotic. And these would be the two sites that we were looking at with that spin labeled side chain. So we decided to see if we can monitor this conformational change to see what the different states look like. And so we used DEER. We had put in 2 spin labels and measured the distance between them with and without globomycin, and these are the distance distributions we got. So with globomycin, and we think based on the KD, we're 100-fold over the KD or so, we have a shorter distance and a longer distance with globomycin. And so what we think this is that we actually have those 2 conformers. One in the crystal structures, we only saw 1 conformer in globomycin and 1 in myxovirecin. But I think what we're seeing is even with globomycin, there's another lowly populated state with the beta cradle and the pH domain. The pH domain sort of tucked down into it. In the apo state, actually, we have it's a broad distribution, but the pH cradle the pH domain also starts coming down into where the antibiotic would bind. And so, for sake of time, I'm not going to go through much of this, but to say that we used Jessica's program to model the side chains and then fit the DEER data for the APO. And indeed, the APO structure looks a lot like the staph with the myxovirecin bound. And then when we look at the globomycin bound conformations, we see that the longer distance, and if we just look at the longer distance, is captured by the globomycin bound structures and not by the myxovirecin structure. So we also collaborated with Phil Stansfeld in England and used molecular dynamics to probe these conformations. So all of this is so that we can visualize what we're capturing in DEER. And what you can see are the C alpha distances between these 2 residues for the APO and the globomycin bound structures. And what I'm doing is taking a snapshot or a structure from each of these regions of the molecular dynamics simulation and fitting it to the DEER in the same way we did the crystal structures. And so then we can visualize different structures multiple structures that might fit the data. And as we get more data, we can refine it. I will say this is a very small conformational change, and these are the 2 residues that capture the change the most. We have a couple more that are more subtle and a little harder to model. And we can use the different structures to again, visualize the different distances that we're seeing in the DEER data. So this is our current understanding. We have an ABO state, which is a distribution of states with a predominantly very closed conformation, that pH domain coming in, and including any of the charge residues in the active site. And then with the single with a single antibiotic bound, we have at least 2 conformations that are consistent with that pH helix, the periplasmic helix coming in and out from that binding site on top of the antibiotics. The antibiotic just has to sort of shift a little bit, and the helix comes down a little further into the groove, and then there's another fully open state that's captured in the MD, but so far we haven't seen in experimentally. We think we might see it in a different membrane mimic, and we've been trying to convince ourselves that might be real. And so I didn't want to share it with you quite yet. So we're in the process of doing some controls and thinking about why we might be seeing that in different membrane mimics. And so with that, I'm going to thank you for your time, and thank NSF, NIH for continued funding. The many people in the lab, so several of the people you saw on the slides are not currently in the group, have graduated, and this is the new crew. And then an amazing team of collaborators that I think I've mentioned along the way. So with that, I will thank you for your attention.
Well, thank you, Linda. Thanks. Well, Linda, it seems to me that this feature of surface loops on beta barrels is common to many of the beta barrels. But you pointed, and I always thought it has to do with protein-protein interactions. But you say that even in the complex, some of the loops are not immobilized in the complex. So do you have an idea of what that's about?
Well, so I think it's balancing two things, which I didn't talk about too much. Neisseria is extremely good at evading the immune system, and it needs to do that by altering its surface all the time. And so the phase variant expression happens with a lot of surface proteins to continuously keep that diverse on the surface. And then the hypervariable region, so this recombination, especially for highly competent bacteria like Neisseria, you'll find sequences I don't know how they do these experiments, but if you have a meningitis infection and a gonorrhea infection at the same time, you'll find meningitis sequences in the gonorrhea sequences. And so it's just about generating as much variability on the surface as it can. And I think it's a mechanism to avoid the immune system. And conformationally, I think it's doing the same thing. It's evolved to have to not have a discrete structure so that it wouldn't be recognized, but yet it's using it to interact and form complexes. And so I think the loops on Opas are actually longer than a lot of the ones I've seen. So they're over 50 amino acids. The two of them are. And I think that's to reach through the LPS. LOS, it's LOS in Neisseria. And so what we haven't talked about is that whole other layer of lipopolysaccharide that the loops have to peek through. Neisseria is LOS, which is truncated. And so I've done, just like I did the planar restraints for the bilayer when we did the NMR structure, I've made one for the LOS as well. And a similar things happens, the loops come closer together, which we saw in the refinement. And with that, only the tips kind of peek out at that point. So, I mean, it's complicated. I don't know, but I think, yes, I think that there is a definite evolved mechanism among bacteria that interact with hosts to have ensembles instead of discrete structures.
There's a question in the chat here from Karen Fleming. Would LPS alter the loop ensemble distributions? It's kind of related to this.
We think so. That's what Megan's working on right now. So Megan's fantastic postdoc that has just been going. I mean, I can't wait to share what she's been doing with you. But she's made all these constructs and has obtained robust expression to the outer membranes of E. coli and now in Neisseria. And so we're isolating directly from there and trying to capture the LOS along with it. At the very least, we can do EPR experiments to look at that. But we absolutely think that, I don't think it's going to change the story. I think it's still going to be an ensemble. I think that's a property of these loops. But I think that it could certainly push conformational populations and the phosphates and the electrostatics near the barrel, I think, are going to really impact the loop orientation. So we hope so, and we think so. And I hope to be able to share that with you soon.
And we'll do another question by Xiaowei Hao. How are the loops in Opa predicted by AlphaFold? I've been getting a lot of AlphaFold questions. I don't know. I'll get to it.
especially after the last few talks and thinking about alternate conformations, we focused a lot on, so the question is, which one did AlphaFold even pick? Because the sequence diversity is so huge. And so I just haven't looked into the database for that. We've been looking at, for LSPA and less about Opa, but evolutionary coupled residues guy, which I think you've inspired us quite a bit to think about. And Rachel Goddett and David Baker. And so, but for the hypervariable sequences, it's just so challenging to think about what could be a conserved structure among them all. And so I guess I should go look. I just hadn't been motivated just because of the sequence diversity that we see. But I'm on it.
And actually, that was a question I was going to ask. Do the ECs give you information about the alternative conformations of LSPA?
Absolutely. Oh, definitely. Yeah, no, it's super cool. It's super cool. And also the ECs gave us, so in Martin's paper, he lists all the sequences and shows the conservation and excludes chlamydia. And then chlamydia, there's one residue that's conserved in every other bacteria, and yet in chlamydia it's mutated. And if you look at the ECs, you find the one that's compensating it immediately. It's in another helix and it's just sticking in there, and it's actually a really cool application of EC. And that's for enzymatic activity. In terms of conformation, there's a set of tryptophans that are or a tryptophan and a phenylalanine that are sticking into the head group region. And they come together when the pH helix goes down, and those are absolutely coupled, identified in ECs. There's a bunch facing the membrane that I'd love to talk to you about because I have my ideas.
Great story.
Yeah,
well,
it's really cool.
Thanks so much, Linda, and thank you for joining the celebration. Thank you.
Thank you.